So I'm going to talk today about something that is relatively new. I usually talk about things that we didn't publish because I'm more excited about this rather than about published things. Plus, you guys can read it already. So I'm going to talk about our studies of physisorption of biomolecules to either flat surfaces or curved surfaces such as nanoparticles. So my group does basically we use a range of different methods, everything from quantum mechanics all the way to actually finite element simulations, but we specialize really in the methods that are based on Newton's equation of motion. So that would be atomistic MD, coarse-grained molecular dynamics simulations, and dissipative particle dynamics, right? So this is our specialty. We use quantum mechanics, again, to parameterize certain parameters in the force field, and sometimes we use finite elements to actually look at the large-scale propagation of the stresses in the materials. Since we look at NanoBio and soft matter interactions, we also use bioinformatics, such as secondary and tertiary structure prediction of biomolecules in our simulation methods. This basically gives a quick overview of several research directions that we work in our research group. Again, we use everything from complete computational biology, which is tertiary structure prediction of transmembrane proteins without known homologs, to, of course, designer solvents, where most of the things in soft matter depends on the solvent. Specifically, polymer nanocomposites are highly dependent on the choice of the solvent, and we're interested actually in the role of the solvent in the morphology formation here. Smart materials by default are very sensitive to the solvent, so that will be the second part of the talk. I will talk about our predictive methods for understanding structure in response of the smart materials. And right now I'm going to talk about this area where we're looking at properties and processes at the surfaces and interfaces, and specifically we're looking at how to optimize the surface in order to get a very distinct response of biomolecules on these surfaces. And again, I start from the flat surfaces and then move on. And another example where we have nanoparticles. So, okay, here. So why do we care about, in general, surface or biosurface functionalization? Again, it's the beauty of this. You can keep the bulk properties of the inert materials, which could be organic or inorganic, and then you add more interesting properties to this, such as biocompatibility, solubility, and of course selectivity. That's one of the most important parameters that we get out of biofunctionalization. Traditionally, you can see in the reviews that people show the covalent or chemisorption of the molecules to the surface. A variety of biomolecules has been already used in this process, such as peptides, nucleic acids, antibodies, dendrimers. And we use this type of surfaces which are functionalized, again, with biomolecules, taking the best out of biomolecules and the best of inorganic surfaces. Traditionally, again, in biosensors, where we can put nucleic acids or, for example, antibodies for the sensing part, or tissue scaffolds, bioelectronics, as you can see here, and of course self-assembly. However, we're very well aware whether we have a covalent attachment of the molecules to the surface or non-covalent, as we see here, the properties of the surfaces and thus devices' performance will depend on the structure of these interfacial biomolecules, right? So our specific structural response to absorption of the surface, again, whether we have it covalent or non-covalent, will change the surface property and thus a device performance. We're specifically interested in physisorption. We use some of the basically covalent approach only to look at the differences. So traditionally, again, you can functionalize, let's say, siRNA to the surface through a linker, and that will be a permanent covalent attachment. Or with physisorption, you use a non-covalent approach where your attachment of the biomolecule to the surface is driven by non-bonded interactions. And that could be electrostatic, it could be van der Waals hydrogen bonds, pi-pi stacking interaction, anything. So for example, here the PEG layer on the gold surface is charged, so the end groups are protonated amines, you can see here, and the negatively charged siRNA can absorb on this surface. However, there's a lot of problems associated with that, whether we're going to retain the base pairs, whether we're going to retain the structures, what should be the optimal surface charge density right here that allows us to retain the properties of biomolecules. Generally, if you're not retaining the properties of biomolecules, it might lead to the degradation of these biomolecules. So anyhow, with Physisorption, we have a lot of positive, or a lot of advantages here. Right? First of all, it's very easy. It has no surface pretreatment. All you have to do is dip material into solution that contains biomolecules. It's not destructive, and it's very cheap. However, the biggest issues associated with Physisorption, again, that we don't necessarily know how the molecule is going to respond to a specific property. So it might lead to conformational changes of biomolecules, which again, detrimental to the performance. And also stability, right? Again, this type of interactions is dependent on the quality of non-bonded interactions, and that means that molecules in some cases can detach quickly and will lose the stability of the surface. So the biggest question in Physisorption, again, what are the desired surface properties that will give you control over this secondary structure or a property of the biomolecule? And that's what we are interested in. For this type of, the first part, we're going to look at DNA molecules, and in general, my group like working with DNA. DNA used in a variety of material science projects. So why do we care? And you guys probably know, but I'm quickly going to say the most important part here. DNA is polyelectrolyte, right? It's really highly negatively charged. Every monomer on the backbone is negatively charged. Plus, it has this molecular recognition properties that are associated with the base pairing. And because of this molecular recognition properties, mostly because of this, we use DNA in a variety of different applications, from, again, technologically relevant smiley faces, or, general, much more complex now DNA origamis, to attaching DNAs to the surfaces for, let's say, sensor development or nanoscale materials assembly. Again, looking at bioelectronic and metallization of DNA. Here we're looking at, let's say we can use, this is actually an example from RNA, but it's the same concept, where we can create a specific fold or even just stretch a DNA on the surface and use a positively charged nanoparticle to get deposited into this structure, which will lead to formation of specific inorganic wires, right? So for example, so again, DNA is used in many different applications. And the beauty of this, that we can have programmed assembly, we can have a pretty good sensor capabilities, as we've been using for the last 15 years in different applications, and also materials with tunable properties. So again, we'll look at DNA and how it's absorbed first on a specific surface. So we want to see, we want to choose a surface that actually is possible to modify in a controlled manner. So not to be very original, we choose the graphene surface, right? And the graphene surface is very simple. It's also, you spend almost no time calculating it. It has outstanding mechanical, thermal, and electrical properties, but most importantly for our study here, that we can controlly change properties in a controlled fashion, change the properties of these graphene surfaces by basically oxidizing it. So again, we can modify the surface chemistry. It is known that once you oxidize the graphene surface, you form epoxy groups and hydroxyl groups, right? So it's a combination of epoxy and hydroxyl groups on the surface, which changes its polarity. Technically, we can go from, again, full graphene all the way to 20-30% of graphene oxide. After this, the structure no longer resembles flat surface, so it's starting to crumble, and you see a little bit not a consistent performance. So using this type of surfaces, again, going from graphene to a 20% graphene oxide, we're going to study how polarity of the surface affects the physisorption of biomolecules. We're going to look at DNA and an example of silk, just for the interest of that. So what we know so far? I mean, we know the first people who thinking of choosing the polarity and controlling the polarity was graphene oxide. This is a recent papers, 2014, 13, 14, that's showing the interactions, simulation specifically, of biomolecules with graphene and graphene oxide surfaces. So you can see in this study, there was a small peptide that had the 3 alpha helices right here. In this study was a 1 long alpha helix, and here was an enzyme with a very specific catalytic pocket. What they found that on a graphene, you see the adsorption, and then there's some loss of the structure, secondary structure of the biomolecule. However, on graphene oxide, we see the adsorption and a general retention of the secondary structure. The same as here, you can see on graphene oxide, we retain the alpha helix. However, on graphene, this molecule is denaturating. If we compare it to this study, it's an opposite effect here, and it's just, again, representative studies. We can see that on graphene, this study shows the enzyme actually retains its structure and stability, versus on graphene oxide, it actually deforms the active site, which means that we have a severe reduction in catalytic activity in here. So there is a general disagreement. There is more papers that shows this type of behavior, that again, graphene denaturates secondary structure, graphene oxide generally preserves it. And there is some studies that also shows that it's an opposite effect. So we decided to join this type of research and try in a controlled manner, go from graphene to slowly oxidizing it to a higher level of graphene oxide and see what happens to our structures. We use atomistic MD simulations for here. For this, again, you guys might be familiar with this, but the best thing about atomistic MD, if you have a decent force field for your biomolecules and surfaces, all you need is a set initial potentials, a set initial particle positions, and temperature pressure, and then all you have to do is integrate equation of motion. And again, your result will depend on the quality of your force field, but in generally it's believed to be reasonable. So the first thing we decided to do, the structure of a single-stranded DNA in solution is still a controversy, and I'm going to talk about it more on the second lecture. But here I want to show that a lot of people actually assume that this is how the single-stranded DNA is going to be in solution. It just kind of sits like this, and then it's going to be adsorbed on the surface. And in additional studies, that's what you see people do. They bring this extended DNA to the surface and then see how it adsorbs. However, DNA, again, single-strand DNA negatively charged, and then we have this aromatic bases here, which are slightly hydrophobic, so they don't like to be extended into solvent. So if we do the molecular dynamics simulations on just simply this strand in solution, and all of my movies are sped up, so they kind of much faster than what it should be. You can see that it folds into these loops structures, which are actually held together through pi-pi stacking and some non-canonical base pairs. So it basically becomes more relatively of a globular shape.
What ionic stress?
0.1 right here. So it's a little bit. It depends on the ionic strengths, but this example will go with 0.1 mole. So what we see at the end, if we're looking at our structure, this is polythymine. So we have again the pi-pi interactions, and we have thymine-thymine base pairing, right? So it's a non-canonical wobble base pair. For RNA, it would be UU. This is TT, right? So this is actually kind of flexible base pair that usually stabilize by 1 to maximum 2 hydrogen bonds. We also looked at, before we chose the lengths, what would be the lengths that has the maximum, not the maximum, it has a reasonable amount of pi-pi interactions and pi-pi stacking. For some reason, my screen, I think, is a little bit bigger. So this is the average number of pi stacks, and I only show here the blue lines, so disregard this part. So here we see that basically up to 15 bases long, we don't have a consecutive pi stack. After this, we have some consecutive pi stacks. And if we're looking at time-in interactions, again, it's a wobble base pair, right? So it has, it can change the conformation. Again, up to 15 base pairs, we don't see much of this time-in base pairing. And after that, we have a certain amount of the base pairing. So we chose for our study actually the 20-mer DNA for this example that I'm going to go through right now, which has some of the consecutive pi stacks and also have a certain amount of base pairing. Generally, our simulations agrees with the force extension curves of single-stranded DNA of polythymine specifically that shows there is some secondary structure formation, which is basically associated with non-canonical base pairing. So we took our pre-folded structure, brought it close to the surface, and here I'm showing two examples. This is graphene, this is graphene oxide. Again, you see a combination of epoxy and hydroxyl groups, about the same as an experiment. As expected, this has been published many times, that DNA, whether we start with extended or folded structure, basically adsorbs on the graphene surface and loses all of this compactness, so it becomes flat. However, on the graphene oxide, we see again DNA adsorbs on the surface. It forms hydrogen bonds here, and then basically retains all these pi-pi interactions that is formed there. So we see that, again, on graphene, single-stranded DNA loses its original folded structure, and on graphene oxide, and the 20% graphene oxide, it actually remains almost intact. It can flip and rotate on the surface. The strength of interaction here is much less as compared to this case right here. We can also quantify that type of process through calculating, again, pi-pi interactions. The blue line right here is DNA base. The red line right here is DNA base surface. And what we see here is, again, on graphene, we lose the DNA base-base interactions, and we pick up DNA surface interactions. So pi-pi interactions on the surface obviously are dominant, and we can see at the end this kind of spreading out. And again, it's pretty typical observation. We wasn't surprised by that. For the 20% of our graphene oxide, if we can look at the base-base interactions, we basically retain most of them, and also we have retain just one actually pi-pi interaction formed, and it's not increasing either. So this simulation is, again, a relatively small system. It was 100 nanoseconds. We didn't see a change in a trend. So next thing, we decided to look at the variation, again, between graphene and graphene oxide. And we, in controlled manner, changing the level of polarity on the surface. Here we're looking at how well it spreads on the surface. So we're looking actually at the density profile of DNA along the z-axis away from the surface. So if it's flat, as in the case of graphene, you're going to see the density, maximum density is close to the surface, and that's what we see right here. And if it's extended, such as in this case of graphene oxide 20%, we see this gray line, right, that has a much higher density away from the surface, and it's calculated over the whole trajectory. What was interesting for us in this case, that if we have 5% of graphene oxide, we see a shift in the density in a way that we retain some of this structure, but really few. And then we have very similar profiles between graphene oxide 5 and 15%. So it's a different regime as compared to just pure graphene. And again, changing it from 15 to 20% led to another shift right here, whereas this DNA now is only participating with hydrogen bonds with the surface and retains its pi-pi interactions. So we have 3 different scenarios in this case.
Question. You have quite a bit of bowing in
Yes.
the graphene oxide. How are you measuring
From the center of mass 90 degrees towards the surface. So even if it's moving, we take the center of mass of DNA and we calculate it with a vector 90 degrees to the surface. Interacting with. What?
Why does it best?
It's lost some aromaticity, right? So you put 20% of epoxies and hydroxys groups, you kill the aromaticity. If you go higher, it's actually going to crumble even more. So if you go higher than 20%.
You don't have boundary conditions.
No, we don't have it. It's actually free floating right here. Yeah. It's a free floating, thank you. It's a small flake. We also wanted this to happen actually because we wanted it to flex a little bit, so it wasn't on purpose. Thank you for pointing it out. Okay, so the next thing we decided to see how different it is in terms of the properties. One of the properties that we like the most is persistent lengths. It's a basic mechanical property, basically shows you the stiffness of the polymer. It's very easily estimated from MD simulations, right? All you have to do is calculate vector correlation analysis throughout the backbone. So it's relatively precise in terms of, again, getting this information from MD simulations. And it's also known from experimental perspective of view that we can impact mechanical properties, electrical conductivity, all kind of different things by changing the persistent lengths of the molecule. So here's the measure of the persistent length. This baseline here represents approximate persistent lengths, again calculated by us, of the DNA in the solution. And this is oxygen coverage of the graphene. So this is pure graphene, this is 20%. And this is actually different sequences, so we were looking specifically as polythymine in this case. What we see is generally linearly decreases as you increase the oxidation coverage. You see the linear decrease in the persistent length until it approaches approximately the persistent length of the original or DNA in solution, right? So that shows us that even though 15% on the previous slide shows us that we lost some of the secondary interactions, we still retain the flexibility similar to the one received in solution. So basically, high levels of oxygen let us retain the single-stranded DNA fold and its basic mechanical properties. So then we decided to see if this is going to be the case for much stiffer molecules, such as double-stranded DNA. We took a very short segment on this. Why did we do this? We wanted to have the exact comparison for the number of bases, right? So we had 20 bases for single-stranded DNA. We have 10 base pairs for double-stranded DNA. And again, if we I think I did not animate this. And if we look at the graphene versus graphene oxide, again, these movies are highly sped up, right? So it's skipping a lot of steps. You can see that the double-stranded DNA also tries to form pi-pi stacking interactions on the graphene. However, in this case, we also retain some of the base pairs. And for graphene oxide, again, the DNA is strongly bound through hydrogen bonds to the surface of the graphene oxide and retains most of the base pairs. However, it loses its helicity. So this high oxidation level of graphene oxide is actually also not maintaining the helicity of double-stranded DNA. So if we're comparing it here, again, this is the height distribution. We can see that in case of double-stranded DNA, again, it's spreads stronger on the graphene than graphene oxide. However, the difference here is less than in the case of single-stranded DNA.
How large do these aromatic islands need to be in order to still see
Pi-pi stacking interaction? 10%. So that's 10%. So it would be. So you can see here, you will also have, like if you look at right here, there's still some islands of aromaticity, but it's not enough. So it's usually only can form like 1 or 2 pi-pi stacks, and it's not mobile, so it cannot move once it's attached to the surface because it's steric hindrance. So with 10%, it can still form more than 2 pair 10 mer. So it's if we compare the non-bonded interactions, which one interacts better, single-stranded or double-stranded DNA with graphene versus graphene oxide? So black here is non-bonded interactions within the DNA, so that's all of them, base pairing, pi-pi stacking, and hydrogen bonds. And the red one is interactions between DNA and the surface, and this is pi-pi stacks or hydrogen bonds, or both, a combination of that. We can see again that for this is a little snippet of the simulations for single-stranded DNA. Originally, we lose, again, the reason I'm showing the beginning of simulations, because here we see some kind of change and then it stabilizes after that. We see that we lose some of the strengths within the DNA, within the DNA interaction. Sorry, I'm showing the wrong curve. And then the DNA basically retains the rest of these strengths and also has interacts stronger within itself than with the surface. For graphene, of course, it interacts much stronger with the surface than with itself, as we can see here. For the double-stranded DNA, we can see here we have a competition between the strengths within the DNA versus strengths within the surface, and that leads to the loss of the helicity. The 20% oxidation level is too much for double-stranded DNA. We'll lose the helicity here. For graphene, again, we see a difference as compared to single-stranded DNA, but again, it interacts stronger with the surface than within itself. So this means, again, if we're talking about the strengths of absorption on the surface, the graphene would be a better choice for both single-stranded and double-stranded DNA. However, with the oxidation levels, will control the retention of the pi-pi stacking or some of the helicity. As compared to just simply Physisorption.
So I noticed that you only have 3 nanoseconds per dsDNA. Is that because it happens much faster?
Yeah, no, it's just we wanted to highlight this jump right here. Otherwise, you can see it, and you're going to see just a flat level. So we just zoomed in the beginning. So it's a zoom-in version. It's 100 nanoseconds, both of them. All right, so overall, we see that, again, the nucleic acids, both single-stranded, double-stranded DNA, tend to retain some structure on the high level of oxidation, such as specifically 20%. And we were starting to wonder whether this 20% is something that of interest is. There is a specific magic number for the biomolecules. So we decided to actually, we didn't decide it. Vladimir Tsukruk from Georgia Tech, he's an expert in creating the composites out of graphene oxide and silk. And he also noticed a very similar behavior as we did with this graphene oxide behavior, but for the silk structure. So in his case, again, he showed that if you combine the Bombyx Mori silk of about 3,000 amino acids long, he shows the high improvement over the modulus and toughness. The hypothesis for this improvement was that silk contributes to some kind of interactions with graphene oxide, and which leads to a better composite structure. Moreover, they compared silk to silicon dioxide right here. So it's experimental again, it's not our studies, right? And for silicon dioxide, again, because they thought the morphology and secondary structure of silk is some kind of contribution to this type of interactions, they took a more polar surface, right? Silicon dioxide is pretty polar. What they saw that the random coil, which is disorder, actually is higher on silicon dioxide as compared to graphene oxide for silk. And also they see that there is a higher percentage of secondary ordered structures, such as alpha helix on graphene oxide, as compared to silicon dioxide, right? So it's contrary to what we were thinking, that the higher the polarity, the better it is for this retention of the ordered elements of the structures. So they believe that the surface of graphene oxide might promote crystallization. And there's a magic number, which is, again, specifically a high level of oxidation, 20%. 10% didn't work. And that's level of oxidation, this higher ordered fraction of ordered elements, which leads to a better mechanical properties. So this is basically a simulation setup of what we used. Again, the silk structure has a combination of a crystalline and amorphous sides. We chose specifically about 260 amino acid segment, which has of Bombyx Mori, which has the same distribution of crystalline and amorphous regions or crystalline amorphous sequences as in the whole Bombyx Mori. We use the bioinformatics to create the first actually structure. Well, it's easy because, crystal structure of crystalline silk is there. So basically we created this long structure and deposited onto the surface. And again, we studied 3 different surfaces: graphene oxide, which has a combination of epoxy and hydroxyls, and silicon dioxide. For silicon dioxide surface, technically you have 2 different contributors here. We have silanols, either single or germinal silanoids. So basically that's the same as hydroxyl groups here. And then we have siloxane bridges right here, which is very similar to the epoxy groups here. So this type of, again, similar surface structure in terms of flatness. However, also we have here many different, basically all the siloxane bridges are the majority of the structures, and it means that everything is covered with basically epoxies and there's some hydroxyl groups on it. So it's much more polar in general. We did expect a similar response to these 2 structures. And if we look at this structure, so this is our initial structure. It had a high level of crystallinity. This was amorphous region, and this was amorphous region. On graphene, we again see this spread, but in general, maintenance of some of the secondary structures. And then graphene oxide versus silicon dioxide has almost similar distributions. If we look at the diffusion coefficient of the silk on the surface, again, graphene, the silk basically slides around the surface, and graphene oxide and silicon dioxide has almost the same sliding force. We also see that if we're looking at the height distribution, silicon dioxide and graphene oxide also have the same one. So we don't see any difference here. We also don't see any difference in terms of non-bonded interactions in the way that we overall, compared to graphene, the silk actually binds stronger to graphene oxide and even slightly stronger to silicon dioxide. The distributions here, this is electrostatic, and this is van der Waals interactions. If we calculate the internal bonds on graphene versus graphene oxide, silicon dioxide, again in graphene we see the slight denaturation of the silk, and it's going to continue if we show it more. For graphene oxide and silicon dioxide, we still see primarily maintenance of the secondary structures. And then we see the interactions with the surfaces generally increase, and they're going to continue flatten out about 100 nanoseconds. So again, we don't see much of a difference here. Slightly different distributions, but on average is the same. What we saw as a difference is actually interactions of backbone versus side chain for graphene oxide versus silicon dioxide. And this is the biggest difference within how the silk responds to these 2 different structures. So for graphene oxide, backbone forms more hydrogen bonds with the surface than for silicon dioxide. And here we have more hydrogen bonds formed with the side chains. We know that backbone and proteins is the most important in terms of forming local structural parameters which control the overall protein folds. So what does it mean for us if we compare? So we can see that backbone actually forms more hydrogen bonds with the surface. If we look at all of the secondary structure analysis on the surface, we see that here is the average or change in secondary structures. This is 0. Positive means more secondary structure, negative means less secondary structure. We can see for graphene, this is disorder, so it's coiled, so the protein denaturates. We also see on silicon dioxide, protein denaturates, more coiled structures are formed. However, for graphene oxide, we see that again, disorder is, or coil structures are decreasing, and secondary elements are increasing. All right, so, yeah.
How much do these patterns change? How much do these results change with the change in the pattern of oxidation?
It does. 20% is actually a magic number. So five.
Pattern, I mean, like, where you have the epoxy group versus...
It's random distributed, right? So we do it absolutely random.
So if you take a second simulation with a different random distribution, how much would these histograms?
Oh, each of these simulations repeated 3 times for the different random number. So it's, it's based on the 3 different surfaces, basically, with the same oxidation level.
How does it vary between different?
It has a standard deviation, but the trends are the same. That's why we put the change in secondary structure instead of like a real number. And this is actually the best representation, right? So for example, this is one of the surfaces that showed a more clear trend in terms of, again, disorder is decreasing on graphene oxide and order is increasing with time in graphene oxide. But generally, the trends are the same for all 3 of them. So we see that actually for graphene oxide, again, we have an agreement with, as compared to silicon dioxide with the experiment. We don't have the same formation of the alpha helices. Actually, we have more beta sheets formed here, but that's actually probably related to the quality of the force field. Because we have some of the increase in the alpha helix, which is this orange, and you can see slightly more than for silicon dioxide, but not as much as beta sheets and beta turns. All right, so that was our major contributor. So in general, we got an interesting results. We also see with this time frame that actually we are recovering. So compared to our original structure, we see the rearrangement and recovery of the secondary structure elements. That's why it's increasing. So we have the interactions with the backbone with the surface leads to recovery of this type of elements. So the only difference, again, we saw between these 2 interactions, the secondary structure is very different because, again, the way the backbone and side chains interact with the surface, and that relates to the distribution of the polar groups on the surface. Okay, so again, what we found, graphene has very strong binding with biomolecules, but secondary structure is not retained. Polar surfaces generally better for secondary structure retention, and we found that it has to be a Goldilocks rule, so that means more investigation has to be done into other different surfaces to see what means not too polar, not too hydrophobic, right? So again, very polar surface such as silicon dioxide also was not very good for secondary structure for silk, for example, whereas hydrophobic surface is also not good. The person who did this work is Ho Shin, and he's continuously working on improving that. So we're looking at more surfaces and more biomolecules to get a better idea whether it's a general observation or observation only pertinent to silk and DNA. Okay, so the next part, and I'm talking too slow, right? I have to pick it up. Too many things. All right, so the next part is looking at DNA and interactions with nanoparticles. And so there's 2, here we actually only looking at the modification of the surface charge of the nanoparticle and see whether we can get the bending, which is important, or wrapping, which is important for drug delivery, or we can get a stretching of the DNA, or decoration of DNA, which is important for example, bioelectronics. So again, we're trying to design a nanoparticle which will give us a very specific response. If you wrap the DNA too tight on a nanoparticle, the enzyme will degrade it, so it has to be just right. The question is, what is just right? The just right would be the same as on the histone protein, right? So that would be just right. So we should have exactly the same DNA conformation, which retains all of the base pairs and also has a very specific tilt and roll parameters as we have it on histone and on a nanoparticle. We also would like to have no denaturation when DNA or anything else adsorbs on this nanoparticle. So here we have the histone protein is about 10 nanometers in diameter, so we choose a nanoparticle with about the same size, 10 nanometers in diameter. Jessica and Abhishek worked on this. So this is our nanoparticles, and they can be small, and I'll start with small and show then a little bit larger nanoparticles. This nanoparticle was chosen again because of this hydrodynamic radius. It's also Vincent Rotella has the experimental data available for that particular nanoparticle, so it's easier for us to compare. He has only one charge, plus 48. This nanoparticle has 1.5 millimeter gold core and 135 gold atoms, has 60 ligands. And then we modify the end of these alkyl chains from traditional alkyl chain, right, so hydrophobic, to a charged one, protonated amine. We also see that as we modify the charge of the ligands, again, ligands are hydrophobic, so in polar solvent, you're going to see the pack up like this, right, so this is going to be a change in shape, and as you increase the charge on the end groups of nanoparticles, you're going to see the change in the shape, overall shape of the nanoparticle. The potential that we chose here for the gold is all harmonically bonded, so it's not a Lennard-Jones, and the gold is not charged, not reactive, but retains all the degrees of freedom, so it can migrate throughout the everything. So as you can see here, it rotates and moves around. So the core of the gold is not fixed. Nothing is fixed here. It's again, we're looking at different levels of the salt concentration. This is 0.1. Here, we have plus 18 charge. This is plus 30, and this is plus 60 nanoparticle charge. We can see that the ligands again behave differently. All right, so we see the packing of the hydrophobic core of the ligands again, and we see that plus 18 does not really do anything with DNA, it just kind of interacts with this weakly. Plus 30 causes local small bending, and plus 60 actually able to bend the DNA pretty well. If we compare this, and we compared it by calculating end-to-end distance of the DNA versus nanoparticle charge, and two different salt concentration. One is, traditional 0.1 mole sodium chloride. Another one is neutralized only. So it's basically a 0 mole and a neutralizing ions. What we see here also, this line represents just the end-to-end distance of DNA in a solution. This line here represents the sodiumer on a histone. It's the same sequence for both structures. We can see that if we have a low charge, the DNA retains its fold. It's all in explicit solvent. Then we have an intermediate structures. For example, from plus 30, we see intermediate folding. And then at certain charges, we see a nice folding of the DNA. So for example, plus 48 seems to be folding it better than plus 60. It's a little bit over compactifying DNA. And in a low salt concentration, it's completely collapsed DNA itself.
So since you're neutralizing it and you have like the 60 there, then you have basically no ions?
Yes, there's no ions, 60. Because DNA would be minus 60, and then you have plus 60. And here we'll have, 12 ions and something like that. So we can see two different regimes, no DNA bending and DNA bending, right? So this is less interesting for us because it's just basically leaves the DNA intact, has a certain binding energy to this. However, this region is more interesting in terms of which nanoparticle is the best that will compactify the DNA in the same fashion as a histone. How do we determine this? Again, we take, DNA has 6 degrees of freedom, right? So the most important ones for the bending are roll, twist, tilt, and twist. The tilt is the most energetic, has the highest energetic penalty in general by itself. So we can compare the histone bending, and then from plus 30, plus 48, plus 60. So this is contribution from tilt on the bending. This is the highest contribution from roll, then tilt. Twist has no contribution to wrapping it on a histone. Then if we combine roll and tilt, we can see most of the contribution arises from here. And the final snapshot shows us that if we compare to histone, the plus 60 actually has a very similar actual bend. So the nanoparticle is very high charge, actually has a similar bend to the histone. Plus 48 has a less bend on the histone. However, if we compare 48 and 60, and now we put two nanoparticles and much longer DNA in general, I just want to see which one will do it faster. So it's a little bit easier to see. Again, it's all atoms. There is no united atoms here. So hydrogens are all present. We can see that plus 60 compactifies the DNA much faster than plus 48. Both of them are successful in terms of compactifying DNA. But again, plus 60 does a much better job in a way that it does it faster. Faster and not necessarily better, though, because it might be that plus 60 over compactifies the DNA. If we look at DNA versus RNA, the difference is the persistent ranks, basically, and the helical rise. So it's much stiffer molecule. And we look at the same end-to-end distance contributions. We can see that if we have it in physiological pH, at a 0.1 mole sodium concentration, the RNA does not bend at all. So only has no salt. And if we look at DNA versus RNA, this is salt concentration versus nanoparticle charge. Again, we see that RNA doesn't have a nice bending, even though some people believe that you can really nicely wrap the siRNA on the nanoparticles for the drug delivery. We don't see any of the nice smooth bending, which will basically not allow the enzyme to digest the RNA. We don't find any parts here that allows us to do the Physisorption of RNA on the nanoparticle. Depends on the charge. Also, I'll just skip this, I think, and show you that we noticed that more spherical nanoparticles lead to much better folding in general of the DNA, much smoother folding. So the shape of the nanoparticles are important. So it's based on nanoparticle anisotropy. So because of this, we decided to look at two different cases. This is with PEG ligands, more hydrophilic, so it makes it more spherical particle. And this is just much larger core. So we increase the core and reduce the length of the ligands. Apparently, if we have a very large core, even though the charges are the same, the hydrodynamic radius are the same, but the flexibility of the ligands are reduced because they're shorter, we cannot bend the DNA as well. So it does not want to bend on a large nanoparticle. I am just going to stop right here because I have too many things anyhow. I'm just going to skip this all and go right here. So we also looked at the nanoparticle concentration versus nanoparticle charge. We found 3 different regimes for DNA. Here we actually have damaged DNA, the DNA with a very small charges and hydrophobic ligands denaturates upon adsorption. So it's again Physisorption. Here we have a nice smooth bending of DNA, and then the largest region here where we have basically DNA structure is still straight. Either we have a very low charges or we have high charges and high concentration. It stretches the DNA in between the nanoparticles. So overall, since I spoke almost an hour, I don't know how that happened. I'd like to thank my group. Again, people who worked on this is Ho-Shin. He worked on, again, he's primarily working on the surfaces and biomolecules. And Jessica and Abhishek, who is a postdoc in my group, they working on the nanoparticles interactions with, again, designing the nanoparticles for a specific response of biomolecules. And our funding, of course. And thank you.